Good morning, good afternoon, good evening, depending where you are connecting from. And welcome to this last for the season Redtalks. So I'm Katia Basso, and together with Professor Fabrice Jardin and Dr. Sarah Huet, we are moderating today's event featuring Professor Reiner Siebert. I want just to mention that unfortunately the camera of Fabrice and Sarah are not currently working, so you may see their pictures, you will hear their voices, but you may not be able to see them live. So let's go started, and it's my actually privilege to introduce today's speaker, as I mentioned, Reiner Siebert, who is a professor of human genetics and the director of the Institute of Human Genetics at Ulm University and Ulm University Medical Center in Germany. Professor Siebert obtained his medical degree from the University of Essen in 1997, and he further pursued his medical and academic training under the mentorship of Professor Grote and Professor Schlegelberger at the Christian-Albrechts-University in Kiel. Here he was appointed to full professor in 2006, and later in 2016 he moved to his current position in Ulm. Professor Siebert, besides being the medical head of clinical laboratory providing genetics service, pursues a very broad research agenda that focuses on the genetics and epigenetics of hematologic malignancies and pediatric cancers, but also of other disorders such as obesity, imprinting syndromes, and neurodegenerative disorders. His team has actually contributed to a very relevant consortium, including the Human Genome Mapping Consortium, the International Human Epigenome Consortium, and the International Cancer Genome Consortium. And Professor Siebert himself is a fellow of the Center of Integrated Quantum Science and Technology. All these contributions actually underline the commitment of Professor Siebert and his team to the mapping of the normal and disease-associated genome and epigenome, and the identification of biomarkers and development of new genomic and epigenomic assays and technologies. Professor Siebert and his collaborators have contributed extensively also to the study of B-cell lymphomas, trying to integrate clinical, genomic, epigenomic, and transcriptomic data. And in particular, he presented cutting-edge studies on the molecular characterization of Burkitt lymphoma, which have been published in prestigious journals like the New England Journal of Medicine and Nature Genetics. Today, Professor Siebert will tell us about MYC-translocated leukemias and lymphomas, from multi-OMICs analysis to pathogenetic concepts. Professor Siebert, the stage is yours, and we are looking forward to your presentation. Thank you.
Thank you, Dr. Basso, for this very, very kind introduction. Thank you also for the organizers for inviting me to give this lecture. Thank you all for your attendance to listen this morning, this afternoon, this evening to this lecture. My aim is today to provide you a bridge between current and former multi-omics analysis and probably old or new pathogenetic concepts in the development of MYC-translocated leukemias and lymphomas. But before doing so, I would like to outline that, as Dr. Basso mentioned, I'm from Ulm University, and Ulm is the city where Albert Einstein was born. And so I would like to take the opportunity also to quote the one or the other important sentence of him, which probably could be also teaching for what we are doing in lymphomas. Additionally, I would like to state that my talk today not only gives you like novel research findings in part unpublished, but also will go back like 30 years in history or longer to some former studies, a bit in the direction of what Sidney Brenner mentioned, that current scientific research should be either 6 months ahead of a bulk or pack of researchers or 30 years after them. So I probably mentioned a couple of studies which have long been known, but probably are forgotten at the moment. My overall view on lymphoma development and lymphoma omics is that it is absolutely hard to map a given lymphoma. At the end of the day, each lymphoma maps to a space of omics data, which on the one hand is the development of genomic operations starting from the germline, and I will mention a couple of issues on germline genomics in lymphomas, via somatic changes, driver events, and then clonal progression. On the other hand, we have a huge number of genomic and omics level operations, from DNA to DNA modifications to protein function. So all this together and mapping the different diseases here, we have to try to map each separate lymphoma in a kind of space. And we have tried this already we have started almost 20 years ago in the German network Molecular Mechanisms in Malignant Lymphoma, in which we have meanwhile profiled more than 1,000 lymphomas by transcriptional and genomic means, and we're still continuing to do so. And when you map all these lymphomas, and we focus mainly on germinal center-derived lymphomas based on gene expression, you will see that they form a kind of network or room, and that's not, I have to emphasize, with single cell analysis, that's just transcriptional bulk analysis. But what you can see is that Burkitt lymphomas map to the top of these network, and if you want to show it in a different space, they show a separate landscape of transcriptional operations, which you can differentiate from other lymphomas like diffuse large B-cell lymphomas, follicular lymphomas, and many others long since known or recently identified like the 11q lymphomas. So if we start to talk on IG-MYC-translocated lymphomas, we have to mention the studies back in the early 1970s when chromosome banding was developed. And that's probably the first chromosomal description of an alteration in Burkitt lymphomas. That's the structural aberration of chromosome 14 described by Manolov and Manolova, which turned out to be an 814 translocation, as shown later on by Lore Zech when she introduced chromosome banding analysis. So there's a whole number of landmark studies by Dr. Dalla Favara and others, who then identified the causative genes and showed that this translocation leads to altered expression of a MYC oncogene due to translocation to regulatory elements of immunoglobulin locus, a mechanism nowadays called enhancer hijacking, at least in solid tumors. If you further continue with genomic studies, then in the early 80s, it was shown that these translocations involving the MYC gene do not only occur in Burkitt lymphomas, but in various subtypes of other lymphomas, mostly derived from the germinal center, but also, for example, in multiple myeloma. And since then, that was the beginning actually of those studies, which we nowadays would likely discuss as studies on double-hit lymphomas and others. Taking all this knowledge of the past 30 to 40 years together, and probably much too easy model could be that the typical aberration in Burkitt lymphomas is the IG-MYC translocation as an initial event, and I want to discuss with you later whether this is really an initial event, followed by a few additional events, and usually the karyotypes of Burkitt lymphomas are not that complex. and only in progression you have the complex karyotypes. Nevertheless, the translocation IgMYC also occurs in other diseases, and when we did our gene expression studies based early in 2000, we developed, like others did, a gene expression profiling characteristic profile. And at that time, we defined what we called core Burkitt lymphomas here in light green, which showed the typical histology, the typical genetics of Burkitt lymphoma. And you see there are quite few, because many Burkitt lymphomas show the one or the other operation. They are similar to Burkitt lymphomas, but do not show the typical histology or others. And what you also see is the genomic complexity, which is low in these core Burkitt lymphomas. And then we did a core group extension analysis, defining a gene expression profile of Burkitt lymphomas and contrasting this to non-Burkitt lymphomas with an intermediate group here, which is a kind of wastebasket of cases which also have MYC translocations, but the dark green shows that many of those show a MYC translocation to a non-Ig partner. That's almost now a 20-year-old study, but we still use this gene expression profiling to define what might be a true Burkitt lymphoma. In addition, some key conclusions can be drawn here. On the one hand, that there exists few cases which look like Burkitt lymphoma, which do not have a MYC translocation, and we nowadays know that these cases are predominantly enriched in lymphomas with an 11q operation, and that there is this intermediate group that at that time was called intermediate group, which is enriched in MYC translocation, and which nowadays contains many of those cases which we would call double hit lymphomas or similar. So putting this additional evidence together, we ended up in a kind of scheme which suggests that in Burkitt lymphoma, the IgMYC translocation is the initial event, whereas in the other lymphomas, like in diffuse large B or high-grade B-cell lymphomas with MYC translocation, the MYC translocation, which can involve either the immunoglobulin locus or non-immunoglobulin locus, is a secondary change, and this is frequently associated with complexity. The question always was, what are the primary events in these non-Burkitt lymphomas? And for sure, there's a huge number of those. And at that time, we defined double-hit lymphomas as those which, for example, have BCL6 translocations and MYC translocations. And I will also come back to this later. What we also showed at that time was that these cases of non-Burkitt lymphomas, so not having the gene expression signature, have inferior prognosis, which was unrelated or independent of classical clinical parameters or also the cell of origin. And that gave rise to a huge amount of research on this MYC translocated non-Burkitt lymphomas. So if we put this simply together, we end up with this scheme. We have a germline in a B cell and probably a non-neoplastic cell population, then an early primary hit and late secondary hits giving rise to lymphomas. And we know that we have two subtypes of lymphoid malignancies with MYC translocation, in which the MYC translocation is likely a very early event. One of them is Burkitt lymphoma, and I will show you later that this is mostly to class switch recombination or somatic hypermutation. And the other one is a very rare subtype of precursor B-cell leukemia, not to be mixed up with a kind of TDT positive transformation of other lymphomas. So a true precursor B-cell malignancy in which the MYC is activated via an apparent BDG rearrangement, and which is nowadays recognized as a precursor B-cell neoplasms in all current classifications. And we have to contrast these to a huge group of other B-cell malignancies in which likely the MYC is activating as a secondary or late event during transformation, and in which frequently enhancer hijacking also occurs to other immunoglobulin, to other loci than immunoglobulin loci. Just one word on these so-called double-hit lymphomas, which have a in addition, an IgBCL2 translocation or a BCL6 translocation or rearrangement. At least the IgBCL2 translocation is supposed to be an early event in the bone marrow cells to occur via VDG rearrangement. So by definition occurs before the mostly class-switched or non-IgMYC translocation. So we can order them already. And this is probably why particularly the IgBCL2 translocated cases of double-hit lymphomas are quite a homogeneous group. Whereas BCL6 rearrangements can occur at different stages and are probably a more heterogeneous group. So these need to be differentiated as it's done by both the WHO and the ICC translocation. And obviously you can argue whether you put the BCL6 rearranged cases in double hits as a separate group or as the WHO classification does as a subtype of diffuse large B or high-grade B-cell lymphomas. But that's more a semantic thing. It's important to distinguish these groups because the IgBCL2 translocated cases, if they do not occur in the lymphoblastic leukemias, are a quite distinct group of diseases. Let's come back to epidemiology. Diffuse large B-cell lymphomas and follicular lymphomas arise with increasing age. They are an age-dependent disease in many instances, and the higher the age, the higher the probability is to get these lymphomas. In contrast, Burkitt lymphomas has been described with a 2 or 3 modal distribution, but at the end of the day, the risk over time is not associated with age, and there might be a peak, particularly in the young age group. If you look simply for the cytogenetic data which is available, then you will see that the single-hit Burkitt lymphomas are strongly enriched before the age of 40 years, and that's based on the Middleman database, so pure retrospective analysis of cytogenetic data, whereas the double-hit lymphomas, for example, are enriched in the older age group. So we have a kind of, if we would put it into mathematics, it seems to be that we have a constant hazard to get a Burkitt lymphoma. It's not that it increases with age. And we have a particular cell population which seems to be present in children and is as a special risk to transform. So one of the questions we have been interested in is what might be the cell population which gives rise to this Burkitt lymphoma, and what are the initial or first hits to drive this. And that will form part of my talk, which is now coming to follow. But one idea could be that the cell of origin of Burkitt lymphoma gives us a clue. Now we have to consider that the cell of origin does not necessarily reflect the cell in which the translocation occurs. Take the example of follicular lymphoma, where it is supposed that the IgH-BCL2 translocation occurs in a kind of precursor cell in the bone marrow, whereas the follicular lymphoma cells itself have a counterpart which is more germinal center B cell. So the analysis of the gene expression signatures gives us an idea of a normal counterpart, which does not necessarily be to be the cell of origin. We have used our gene expression data from bulk analysis, and we did this complex self-organizing map analysis. And I don't want to talk you through this complex figures, which who is interested can read in our publications. But I would like to show you that we can assign certain areas of a self-organizing maps to specific populations which have been identified by gene expression profiling in normal germinal center B cells. And if we lump all this together, we see that Burkitt lymphomas seem to have a specific dark zone signature. And we also see, if we compare the similarity between the gene expression profiling, that Burkitt lymphomas and MYC-negative B-cell lymphomas with 11q operation show a pretty similar gene expression profile, so that might hint to a similar cell of origin. But you can also see that the double hit with BCL2 look pretty similar, whereas the double hit with BCL6 look a bit more different. So it makes sense to segregate them apart. We have followed these studies using HTG-based gene expression profiling in the recent past, and that confirms that the Burkitt lymphomas, in contrast, for example, to diffuse large B-cell lymphomas, seem to have a very constant and very conserved gene expression profile. There might be some slight divergence, but overall, it seems to be a special cell of a with a dark zone gene expression signature, which resembles Burkitt lymphoma. Now, the question is, does the MYC rearrangement occur in these dark zone cells, or is the MYC rearrangement really the initial event in Burkitt lymphomas? And there are several ways to approach this. So we think it's definitely an early event. And we have used modeling algorithms to model the order of genetic operations in Burkitt lymphomas. And I don't want to go into details behind the mathematics behind this, but the bottom line was there seemed to be at least a small subset of cases in which the Burkitt translocation, so the IgMYC translocation, is not the initial event, but it could occur as a secondary event after a kind of initiating event. Is there evidence for this from experimental data? Yes, there is. When we performed whole genome sequencing and transcriptome sequencing in a German cohort of sporadic Burkitt lymphomas, we could see that the IgMYC translocations is mostly class switch or somatic hypermutation related, but we found some translocations which need to have occurred through illegitimate VDG recombination. And that's a process which is normally assigned to the bone marrow and to a precursor of a cell, which then goes to the germinal center and might undergo these kind of translocations. So these data, though a restricted number of cases, might indicate that there is at least a small subset of Burkitt lymphoma which has a kind of precursor lesion occurring before the IgMYC translocation, which occurs before this translocation occurs, and, for example, might deregulate genes which are important for lymphomagenesis, like cyclin genes. There is one other evidence, and that's a study together with Sam Boulatia from the NIH, which is currently under review. And that's a huge epidemiologic study in Burkitt lymphoma. And in this study, it was shown that in cases of endemic Burkitt lymphoma, there might be, or there are chromosomal aberrations involving like chromosome 1 or 8 in normal cell populations of patients with Burkitt lymphoma, and that this so-called mosaic chromosomal aberrations are enriched in children with Burkitt lymphoma as compared to controls. That would argue that patients prone to develop Burkitt lymphoma have a kind of Burkitt lymphoma chip, if you want this, so a clonal hematopoiesis event. That means that there is a mosaic chromosomal aberration which might predispose to Burkitt lymphoma via deregulation of core genes like MYC or 1q genes, which could be driven, for example, by chromosomal instability related to EBV infections or AID activations in subset of cells, and which could precede or given later on advantage to the IgMYC translocation to develop full-blown lymphoma. So Einstein would have said, okay, that has now made the things much more complex. We thought Burkitt lymphoma is a kind of easy model, IgMYC translocation, few other aberrations, but now we are talking on making the things more and more complex. So how to deal with this? The fourth edition of the WHO classification has essentially differentiated endemic Burkitt lymphoma, sporadic and immunodeficiency related. And the WHO classification 5 now suggests, in addition, to use EBV status, because it might better classify Burkitt lymphomas than the geographic origin where the lymphoma occurs. We have dealt mainly with sporadic Burkitt lymphomas, and one interesting thing we found is that many of these Burkitt lymphomas derive through a MYC-translocation in which the partner is in the immunoglobulin locus, particularly in the immunoglobulin alpha region. So that would mean that the precursor cell of a Burkitt lymphoma attempts to have class switching to immunoglobulin A, but this leads to a mistake, and therefore gives rise to a translocation with MYC, and therefore Burkitt lymphoma can derive. That's a hypothesis at the moment, and the hypothesis is that the population at risk is obviously a B cell which wants to make IgA, and IgA is predominantly made in the intestinal B cells, but that's exactly the cells where Burkitt lymphoma occurs. So at least in sporadic cases, we have a high preponderance of Burkitt lymphoma developing in the ileocecal regions, at Peyer's patches, so exactly where the main production of IgA occurs. And if we link, for example, the IgA levels to first exposure to microbia, then you will see that the slope of IgA development in a certain way resembles the incidence which we see in pediatric Burkitt lymphomas. So one hypothesis we have put forward is that the population at risk is a particularly intestinal B cell population, which is prone to develop IgA or poised to develop IgA, which couldn't undergo class switching, and due to a mistake in class switching has the operation of a MYC gene. At the end of the day, the deregulation of a MYC gene is the driving force. And this deregulation can occur on the transcriptional level and leads to an expression of predominantly the translocated MYC allele. Depending on the breakpoint, different splice forms of the transcript of MYC are built. If the breakpoints are 5' of a gene, we have all 3 canonical exons which are expressed, and we have translation starting at the canonical translation start site. If the breakpoints are in the intron, in the first intron, then we have transcription starting in the intron from a different promoter, and we have translation starting a bit later. Whatever the translocation occurs, there is somatic hypermutation, which is shown here in the upper tracks, in the intron, but also in the exonic regions, and the exonic mutations, particularly are close to locations where phosphorylation sites of a MYC protein are, and might lead to the stabilization of a MYC protein. So we have a number of interacting mechanisms leading to the stabilization and overexpression of a MYC oncogene. But MYC oncogene expression alone is not sufficient to drive lymphomagenesis. This has been nicely shown in many mouse models and other studies. You need additional operations, and these additional operations first involve MYC, but otherwise a quite restricted set of genes, with probably the most commonly involved to be ID3. So one thing we looked into is whether these genes are also affected in the germline, which are recurrently mutated in the somatic tissues, and the answer is yes, they do occur, but of course not highly frequent. So germline mutations might be associated with Burkitt lymphomagenesis, and there are of course some immunodeficiency syndromes and others, like here Bloom mutation, which is associated with Burkitt lymphoma. And there are other conditions not yet really investigated, which might predisposed to Burkitt lymphomagenesis, and I have shown you the clonal mosaicisms and other loci to come up, which might be linked to Burkitt lymphomagenesis. Overall, if we look into the landscape of somatic mutations, we see that there seems to be a quite conserved set of pathways which is deregulated in Burkitt lymphomagenesis, one being proliferation and survival, one being the SY-SNF complex, and one being B-cell receptor signaling. And that was nicely shown in editorial by Leas Campo in the Nature Genetics in 2012. Now, one of the mutations we first identified as commonly mutated was mutation of the ID3 gene, which in our hands was seen in like 60 to 70% of Burkitt lymphomas, rarely in intermediate lymphomas, which might still be enriched for true Burkitt lymphomas and hardly ever in non-Burkitt lymphomas. And ID3 interacts with TCF3, and this ID3 is a kind of inhibitor of TCF3, but also of TCF4. And this mutation leads to freeing of TCF3, which then can is kind of activated and bind to DNA. So we were interested whether only genomic mutations can deregulate TCF3 and ID3. And what we saw is that TCF3 can be also deregulated by alternative splicing. And with this alternative splicing changes the binding properties of TCF3, and that this alternative splicing is predominantly seen in cases in which ID3 is unmutated. So it might be an alternative mechanism on the RNA level which activates this pathway, which is also telling us that studies only focusing on genomic operations cannot see the whole space of omics operations, which at the end of the day lead to deregulation of the same pathway. So to explore a tumor entity completely, you need to have all the omics levels together. Another omics level important is DNA methylation, or let's put it more general, epigenomic alteration. And if you again looked at the TCF3 locus, there's differential methylation at the TCF3 locus. TCF3 itself is regulated by TCF3 as its marker 4, and it's highly expressed in Burkitt lymphomas as compared to other lymphomas. And we see the typical pattern we would expect: low methylation, high expression, high methylation, low expression. So there seems to be epigenetic regulation, and that seems to occur at 2 differentially methylated regions in Burkitt lymphoma within the TCF3 gene. So we add another layer of deregulation, that is epigenetic modification, for example, through binding through transcription factors. And I explained to you that for TCF3 we see this typical pattern. That means if you have a high methylation, you have low expression, and the other way around. It's a typical textbook knowledge you would expect. If a gene is hypermethylated, particularly at the transcription factor at the transcription site, the gene is silenced. But what we observed was the totally opposite, and that was quite unexpected for another gene, which was SMARCA4, a member of the SY-SNF complex. So what's the reason for this? SMARCA4 itself it's highly expressed again in Burkitt lymphomas, but in most instances it's not the wild type gene, it's a mutated gene. And these mutations lead to the fact that SMARCA4 is not activated. So what happens actually is that SMARCA4 sticks to the DNA and prevents DNA methylation, but it does not activate its target genes. So that explains why also on a global level, we see a kind of inverse correlation over there. And we came up with this model that in normal B cell, TCF3 is differentially methylated and regulated. It's inhibited by AD3. TCF3 binds to its target genes, including SMARCA4, and that regulates on the genome-wide level other target genes. In contrast, in Burkitt lymphomas, we have deregulation of TCF3 it's hypomethylated, it's higher expressed, it's activated through mutations, or through mutations in ID3, which free the TCF3 gene. That activates its target genes, among them SMARCA4, and SMARCA4 in its mutated form is expressed and again leads to deregulation of a number of target genes. That explains why a very low number of genetic events can lead to a complete deregulation of a transcriptome of Burkitt lymphomas. And if you compare Burkitt lymphomas gene expression or MYC-positive lymphoma gene expression to diffuse large B-cell for lymphomas, for example, you see a kind of avalanche effect. There's a huge number of genes which are deregulated, and you can do a lot of kind of classifiers simply by randomly picking some of the genes because there's such a huge differential expression. If we put the data of many groups now together, we end up with a deregulation in addition to the MYC translocation of a number of pathways. One of them is B-cell receptor signaling. One of them is the TCF3 axis. There's transcription, there is cell cycle deregulation, and there's SWI/SNF complex deregulation, and there is also the GNAR complex and ROA complex deregulation. That seems to be the core event in Burkitt lymphomas. Now, if we conclude at this stage, I have shown you that germline variation predisposes to a subset of Burkitt lymphomas and that these Ig/MYC translocations occur due to mistakes of class switch recombination or somatic hypermutation. There is a skewing of class switch recombination mediated Ig/MYC translocations to IgA and some IgM. That MYC deregulation can occur of many omics levels, like translocation, expression pattern, mutation, and that few other pathways are in addition deregulated. And again, these can be on different layers. So the important message is here, not one layer is sufficient to describe what's going on. And I haven't shown you the data, but you can believe me, a relapse is mostly linear. From the initial diagnostic clone, there are few additional operations with a bit of branching, but not as compared to other lymphomas, we do not see a wide branching of Burkitt lymphomas. You might say, oh, that's complicated stuff. And Einstein might say, if you can't explain it simply, you don't understand it well enough. So let's try to get this into kind of model how our model of Burkitt lymphoma is at the moment, at least of sporadic Burkitt lymphomas. We think Burkitt lymphoma derived from a problem in a primary mucosal immune response. Reason is clinical presentation, for example, and that this might be linked to a kind of germline predisposition, for example, DNA repair or an immune response. We think that this primary mucosal immune response has to be linked to an antigen. And when that there is an antigen which drives Burkitt lymphoma, I do not say it's an antigen coming from the human itself, can be also the first exposure to bacteria or others. And that then the probability to develop an Ig/MYC translocation is increased. Probability means MYC itself is leads to apoptosis. So if there are no additional events, then probably an Ig/MYC clone dies again. And we think there's another Ig/MYC clones occurring in each of us during these events. But only if other alterations are present, like alterations in the pathways I've shown you, then we get a full-blown lymphoma. And if all this comes together, we have a kind of exponential phase. And if you take the doubling time, we think that it's probably 1.5 months from a kind of precursor cell in a fast doubling cell to get a clinical visible lymphoma. And we have again modeled these data, and we think that a total four secondary drivers are necessary in addition to a MYC translocation to model this. That would fit nicely also with the number of core alterations which you need in addition to an Ig/MYC translocation. Let's briefly, that was mainly sporadic Burkitt lymphoma. Let's come briefly to endemic Burkitt lymphoma. and the question, which role plays EBV over there? And I'm coming back to genomics, and there have been a number of genomic studies, and the data of those were summarized by Cristina Lopez in my group in a review. And what you can see is that in light blue are the EBV positive, the number of cases mutated in EBV positive cases, and with yellow are those which are EBV negative cases. And you see that there are differences. Overall, the number of mutations seems to be lower in EBV positive cases, but particularly some core genes like ID3 or TCF3 or cyclin D3 or TP53 seem to be more rarely affected in EBV positive cases. So the question is, what happens over there? If you put together all these kind of studies which have been published and have been comparing EBV positive and negative cases, so the core differentially mutated genes are probably ID3, TCF3, cyclin D3, and SMARCA4. So we investigated DNA methylation in Burkitt lymphomas, and we do this since now more than 10 years. And I just show you some very early whole genome bisulfite sequencing data, which we did in a cell line, to show you the typical patterns. The textbook knowledge, and that is what you see here, is that present transcripts, so genes expressed, which is the green line here, have a low DNA methylation around the transcription start site and a high methylation in the exons. And the absent transcripts have a high transcription methylation at transcription start sites and lower in introns and exons. Of course, transcription start sites is the prototype. You can also look at enhancer sites. But for us, transcription start site is the best indicator of whether a gene is active or not. So we looked at the global DNA methylation in endemic Burkitt lymphomas shown here, as compared to sporadic Burkitt lymphomas, follicular lymphomas, diffuse large B-cell lymphomas. And what you can see here is that endemic Burkitt lymphomas have an excessively high DNA methylation in the mean already, as compared to normal B-cell populations, but also as compared to the other germinal center-derived lymphomas. So we did a lot of sophisticated bioinformatics, and I don't want to go into very detail here, but what you can clearly see is that EBV positive Burkitt lymphomas here on the right side show a strong hypermethylation, and there is a small subset of Burkitt lymphomas which probably has a different latency, which shows also a hypermethylation, but not that marked over here. And if you show this in the UMAP analysis, you will see that EBV status is the most distinguishing feature between these two groups. So here are the negatives, here are the positives. You see there exist some few outliers, but overall that might be associated with latency again. But overall, that's a clear distinguishing between EBV positive and negative Burkitt lymphomas, and that's a kind of independent also whether they derive from Africa or from Europe. So we wanted to know whether these hypermethylation, which we see in EBV positive Burkitt lymphomas, also affects genes which are otherwise recurrently mutated. And without going into too much details, is here showing a number of these genes which are recurrently mutated in Burkitt lymphomas. And here's the methylation status, both in endemic and sporadic Burkitt lymphomas being EBV positive. And what you can see, and that's only shown here for regulatory sites, and for example, TCF3 is included here, or ID3 is included here, that we see a strong hypermethylation at regulatory sites. So what we think is that DNA methylation driven by EBV can affect or can substitute for mutation in EBV positive Burkitt lymphomas as compared to others. Another study we did on DNA methylation is we compared human Burkitt lymphomas to mouse Burkitt lymphomas using a mouse model developed by Sandrine Sander and Klaus Rajewski. But what this study shows you is that the overlap between human and mouse is quite quite low. It's only 22% of the genes which are similarly hypermethylated or hypomethylated from quite an extensive number. So overall, that, at least in our hands, questions a bit the use of mouse models, at least for some of these studies, to describe what's going on in human Burkitt lymphomas. In addition, we shouldn't forget that not only the human genome is methylated, but also the EBV genome is methylated can be methylated in endemic Burkitt lymphomas, and that's something to be explored in the future in how far this affects the pathogenesis of Burkitt lymphoma. So let's try to do endemic Burkitt lymphoma genesis into this model I've shown you before. In addition to the germline predisposition we have in sporadics, we have a kind of malaria predisposition because we know that Plasmodium falciparum is associated, infection, particularly if chronic, is associated with a Burkitt lymphoma predisposition in endemic cases. And this might serve as antigen, but we know that malaria infection also increases AID activity, and that again might activate chromosomal instability and also the MYC translocation. In addition, we have likely a higher number of germinal center passages and EBV-associated silencing of some of the genes, which could contribute this. So in the case of endemic Burkitt lymphoma, we might have an interaction between predisposing factors and both EBV infection and malaria, which helps to develop Burkitt lymphoma. It might explain the higher frequency in malaria-prone countries, at least in some regions. The genomic predisposition in part might be linked to certain populations again. In the final part, I would like to come to some additional thoughts on Burkitt lymphoma, which come from the fact that we also see Burkitt lymphomas in the breasts, and that particularly in bilaterally and in women which are pregnant or lactating. That has been described under the WHO 4 classification as a quite common site of involvement. We did a study in which we reclassified cases from the Kiel lymph node registry, and I have to say, after a rigorous reclassification, the number of cases we see is much lower than the initiating number of samples, because many of those cases traditionally classified as Burkitt or Burkitt-like lymphomas would be nowadays classified as other high-grade B-cell lymphomas. But if you stick with the cases which occur in the breast and in the ovaries, they seem to be younger, and they, as far as the information is associated, particularly in the breast lymphomas, there is this association with pregnancy and lactation. And so let's probably think about what's happening during lactation. So the main product delivered to children through lactation, through the mother's milk, is IgA, as a kind of maternal protecting factor for the child. And you can show that the number of plasma cells producing IgA is increasing during lactation, so during pregnancy and then lactation, and this later on declining. And you can also see this in both in mouse models and in human models. So one idea could be that we have again this IgA theory, which I put forward before, that particularly an intestinal cell poised to develop IgA is the precursor of Burkitt lymphoma. So how does this fit with this IgA in the mother's milk, but also the IgA production in the breast, and how do these IgA cells come to the breast? And there is one study And I told you I will go back a couple of years from 1975, which I think nowadays no IRB would anymore agree on to be performed. But in this study, almost 50 years back, there was a group of females which were pregnant and which were giving laboratory strains of bacteria in their pregnancies. And then there was measured, and this was given orally, and then there was measured the IgA production in the mother's milk and in the mammary gland. And essentially what is shown in this study is that that oral application, so gastrointestinal application of IgA leads to a specific antibody response through the mother's milk. And there are other studies in mice confirming these findings, that obviously to protect the child, there is an intestinal response of the mothers, which then leads to production of a specific IgA to protect the child in the mother's milk. So that would be, again, a factor in which you would say, okay, that could explain why there is the increased frequency of these Burkitt lymphomas in lactating or pregnant women. So I've shown you a number of things which might make the story about Burkitt lymphomas and MYC-translocated lymphomas more complicated. The question is, can we move this to an opposite direction? And a bit provocative, if you put all this data together, my summary would be probably like this. You all know this very well-known formula Einstein developed. And I think the results could pose a hypothesis saying that the epidemiology of Burkitt lymphoma is driven by microbiome to the first exposure and then MYC. We need to identify the cell of origin, really giving rise to this, and then we have an overproliferation which leads to the clinical manifestation. And that's probably a bit provocative, but it gives also food to think and to develop further research agendas. If true, it would be interesting to think about kind of vaccination or so in the future to Burkitt lymphomas, but that's all a bit of science fiction. I have to finish with thanking a lot of people. We have this great collaboration within the German network Molecular Mechanisms in Malignant Lymphoma. And the data I've shown you today have been generated, most of it in this consortia, and the leading persons were Christina Lopez, Shaima Al-Jafari, Selina Glaser, Emil Steinbeck, and Anja Fischer in my group. I thank you for your attention, and I'm happy to take questions.
Thank you, Professor Siebert, for these very interesting seminars. And now the talk is open for questions, so everyone can raise questions in the questions tab, anyone from the audience, and maybe I and our co-chair and my co-chairs will start asking questions. I see no questions right now in the questions tab. So first, actually, a very quick question regarding this Burkitt lymphoma in the breast. Are these EBV positive, negative, and are these tumors actually rising in the breast, or are these cells moving there from some other site?
So the series we investigated were mainly EBV negative. And the hypothesis is that they, okay, so these are mainly primary Burkitt lymphomas of the breast. So that means clinically, we do not have evidence for secondary manifestation. That does not tell you that there's not a micro Burkitt lymphoma somewhere else. But as proposed, we think it might be a kind of precursor which, due to the immunologic reaction evades the gastrointestinal tract and goes to the breast, and that is giving rise to this lymphoma. Hypothesis to be confirmed, but clinically it's mainly primary Burkitt lymphoma of the breast, as far as it could be confirmed.
Thank you.
Thank you very much, Professor Siebert, for this fascinating talk. I also have one question. I was surprised about the enrichment of IgA antibodies, translocation in MYC-translocated Burkitt lymphoma. Do you think the translocation occurs right during the class switch recombination due to the involvement of IgA locus?
Yeah, likely this is the case. That's the hypothesis. Let's do it. So we know that the vast majority of Burkitt lymphoma express IgM. So they do not undergo class switch, active class switching producing IgA, with the exception of probably very, very rare cases which might do this. So we think that it's indeed an attempt of these cells to class switch to IgA, which fails and then gives rise to this translocation.
So it fails for some reason, and then
it fails for the reason that then MYC is translocated to IgA, because if it would have been successful, we probably would have never seen this cell as a Burkitt lymphoma.
And is there any study about the TAD domains, for example, that show that MYC is close to IgA locus, or?
So obviously the breakpoints lead to the translocation of MYC next to IgA. So the activation is on the derivative chromosome 14, and then you have IgA, and then comes MYC. If this is what you mean. So there's in addition, obviously, data from which have recently shown that replication timing likely involves whether this translocation occurs, but this has never been really linked to different parts of an immunoglobulin locus, but only to a very central part in which this was shown. We do not have data, let's see, experimental data, whether you can artificially change replication timing, for example, at the IgA locus, but we see transcription from IgA.
I have a question. Thank you, Professor Siebert, for your nice talk. Regarding this entity, high-grade B-cell lymphoma with 11q aberration, you show in your second slide that the gene expression profile is very similar to true Burkitt lymphoma, despite the lack of MYC translocation. Do you have any molecular explanation based on methylation, as you show, for example, or other kind of explanation?
Okay, first of all, thanks for a very nice question. First of all, you have to say, when we first described these lymphomas, we described them because they show the gene expression profile of Burkitt lymphoma.
Yeah.
That was, I have to thank Harald Stein particularly, who teased me a lot of time and says there are MYC-negative Burkitt lymphomas and your concept is wrong. So we looked into this dedicatedly in cases with a gene expression profile similar to Burkitt lymphomas. Meanwhile, we have extended this to a huge series of 11q cases, and there's still the strong similarity. Methylation-wise, these cases can be nicely distinguished from true Burkitt lymphomas. So we see a different methylation profile, and we see slight differences in the gene expression profile. So our hypothesis is that there might be a similar cell of origin, because we also see a kind of enrichment in younger populations, but there are definitely completely different genomic paths, because the mutational landscape is completely different.
Yes.
The chromosomal landscape is completely different. But what we might see predominantly might be a relatedness of a cell of origin.
Okay.
Methylation is different, to answer your question. Methylation is different.
Okay.
We actually use it as a kind of classifier to map cases.
Okay. And in a clinical point of view, we know that we can cure almost all young patients with Burkitt lymphoma, but in case of relapse, we know also that we cannot cure any patients. It's a huge difference between follicular lymphoma or diffuse large B-cell lymphomas. We know that we can at least treat with a second or third line or fourth line. And in Burkitt lymphoma, it's not the case. We have one shot. Do you have any the opportunity to understand the mechanism of resistance, chemo resistance, for example, for in this situation?
I would be happy if I would have. So we have compared probably a dozen of pairs we had in hands on the genomic level and DNA methylation level. We see this kind of slightly linear evolution, but
example,
the different
P53 or no?
Yeah, of course, P53, that's known as a prognostic factor. But overall, the differences between relapse and initial diagnosis are not very strong. Methylation, with regard to methylation profiles, we failed to identify a relapse sample from initial diagnostic sample, and we failed to identify a DNA methylation signature which differentiates relapsing, at initial diagnosis, relapsing Burkitt lymphomas from non-relapsing Burkitt lymphomas. So we have no marker really in hand except TP53, which is as described as prognostic factor. Additionally, it seems not really to be a kind of relapse. It's mostly progressive disease. You have it very early, you have it very rapid. There seems to, yeah. So we yet have failed. Katja Basso might have the things in hands, but we don't have them yet.
Well, we are working on it. So there is actually a question in the chat, so I will read it for you. I think you cannot see it. So it's from Emmanuel Bellard, and asking, after listening to your point on mouse models, can we still trust mouse models, more particularly those trying to demonstrate the contribution of additional events in the onset of the lymphoma?
Oh, difficult answer. I think yes, it's the best one we can do. Let's say I always emphasize that I'm a human geneticist. That's my job description. So my focus is on human disease. We were interested in how similar on the methylation level these lymphomas are, and we did different analysis also on other types of lymphomas. And I have to say, at least from the methylation level, there are not that many similarities. So we were astonished. We were expecting that we have very similar things, but we don't have them. But that's only one omics layer. It doesn't tell you that on the other omics layers it doesn't work. But at least the overall idea, probably the overlap is what's really key. That's something we don't know. And the other events are probably secondary and not important. That's something we need to understand. But overall, we were yet surprised by the not extensive overlap between DNA methylation data of both species. They are different, they have different immunologic system, but there are great studies on mouse models, and there would a lot of our current understanding would not be possible without these studies. I think they have their value, but we need to interpret it on the respective omics level with caution.
There's actually a little follow-up question. Is always about not human genetics, so I don't know if you want to address it, but the question is, what about dogs? Would those models be similar regarding methylation? I'm not sure you have looked in dogs.
I actually, and incidentally, we have been submitted a dog lymph node for diagnostics, and we were wondering about the name, but that's my only and solitary experience with dogs, so no comment from my side on this. I have no experience.
So while the audience eventually had new questions, I wanted to ask you about, commenting on this. So as you very well pointed out, practically the translocation of MYC, mostly in the IGH locus, is really a hallmark of Burkitt lymphoma. But we know very well that is not enough. So you need these additional events, again, that you described very clearly. So how do you think this translocation and therefore deregulation or aberrant expression of MYC is strongly linked, right, with this germinal center cell of origin that you were discussing? So if you want to comment on why MYC is essential, although it's not enough, that's the point behind that.
Because it likely, so first of all, it's obviously a well-known oncogene which has oncogenic properties. It just needs to be, it needs to have a rescue, to have a breaks off from the other parts of the cell. And the interesting thing is that if you look at its targets, it likely activates a lot of these genes which are all cooperating and then allows somatic hypermutation, which can occur in the germinal center, like on ID3. We know that a lot of these ID3 mutations are likely linked to somatic hypermutation. So this you get if a gene is transcribed. If MYC switches is on, then it probably helps to have this helper mutated. So I think it's a complex interacting network. And MYC induces a lot of genes, not only those which are helpful for MYC transformation, but also the other way around. But it seems, at least in the germinal center, it has the environment that machineries like the somatic hypermutation machinery might help to take the brakes off.
Thank you. Any other questions from audience or the other chairs? Okay, then if not, I take advantage, but please feel free to interrupt me. Can you comment on eventually biological differences between pediatric and adult Burkitt lymphoma? We know, right, that in the children population, actually, Burkitt lymphoma is the most frequent B-cell lymphomas. So you mentioned predisposition and so on, but can you comment a little bit more?
Yeah, I would say there's likely two comments. First of all, the adult population has a slightly different mutational landscape, more associated with EBV. And SOX11 expression might be different, so there might be slight differences. The other point is that you need to be careful in a lot of epidemiologic studies which have not applied current diagnostic criteria, so they might be mixed up, like we have seen in the breast lymphomas with other high-grade B-cell lymphomas. So particularly the old or elderly epidemiologic studies showing some peaks in the old age group need to be curated for this. And the third thing is, if you believe or not in the IgA theory, then obviously you have kind of senescence, so these clones, these IgA clones might die certainly. And if you take, for example, immunodeficiency or treatment with immunosuppressing drugs, so after transplant or some infections, then you might lose some of this memory of the IgA cells that makes you to react again on the microbial exposure. So it could happen then that you have lost such a clone, you need to have it again with a special response. And that means that's what I mean. There is a constant risk that is increasing in childhood, because there you have to grow all this IgA clone, but it's never fading completely, because you will always have new exposures, and probably also some dying clones you need new responses on. And therefore we have the old adult group. It might, as said, be linked more to immunosenescence, immunodeficiencies, and therefore also probably to EBV, but that's work to come up.
Anyone else up there?
Ryan, you don't mention MYC amplification in your talk. Is it a frequent event, or it's a marginal event in B-cell lymphoma in general?
In B-cell lymphoma, it obviously occurs, and we see it regularly. And you see also this kind of complicons where you have amplifications in what we formerly called homogeneously stained regions or double minutes and so on that exists all. But we don't see it in Burkitt's. If you see it in Burkitt's, you would ask it's the Burkitt. But in other lymphomas, you see it. And you can see it together with and without translocations. And it's one of the factors I would judge predominantly as a factor which is a secondary event which occurs during further transformation progression.
Okay.
There is one more question from the audience. Gail is asking regarding the Burkitt lymphoma curation, I'm not sure if it means treatment, would the examination of the Burkitt lymphoma gene expression signature be sufficient for discrimination?
You have to repeat, I think I didn't get it completely.
So, regarding the Burkitt lymphoma curation, would the examination of the Burkitt lymphoma gene expression signature be sufficient for discrimination?
I think I cannot tell you because based on the limited data we failed yet to do. I think that needs larger efforts to put together really relapsing diseases and patients who relapse and do it on a broader level. So everybody of us has only a subset of cases which is really relapsing under appropriate treatment. And therefore, we are all yet in a kind of underpowered to really do this. I would say the book is not closed on this, but yet at least our attempts failed to identify a prognostic signature.
So, no more questions from the audience. We have still a couple of minutes if there is any curiosity or I will ask you a last point then. You were actually showing some of your data about EBV positive and increased DNA methylation. Do you have any thought about the connection, direct connection of EBV in impacting methylation process itself?
Yes, we have, again, okay, DNA EBV infection affects the, at least in what we see is that there might be a link to the ubiquitination system, and then via this also to the TP53 activation, but also DNA methyltransferase activity. So you can, there is evidence linking some Epstein-Barr virus components to DNA methyltransferase activity, probably via ubiquitinin, and that's something we are exploring at the moment. The other thing you have always to consider is that also it might be a germinal center passage, because in the germinal center we see a lot of methylation going on.
Sounds good. I think with this question we have to close the session. And I would thank everyone for attending and Fabrice and Sarah to co-chair this session. And of course, our many thanks to Professor Siebert for sharing with us this work. Thank you. Have a good day.
Thank you very much.
Thank you very much.